We're back. We're back to finish meiosis cell division. We started with meiosis 1. We had a cell with 4 chromosomes. Its diploid number was 4, and it went through meiosis 1. We ended up with 2 cells at the end of meiosis 1. Their chromosome number had already been reduced, cut in half, so that each cell at the end of meiosis 1 has 2 chromosomes in it, one long one, one short one. This one happens to have a long green and a short red. Remember, I also haven't drawn in these pictures, I haven't drawn in the effects of, or the results of crossing over or genetic recombination. So some of these sister chromatids might not look like each other because they have little pieces from their homologous partner before they separated. Okay, so now I want to draw what happens to this cell as it goes through meiosis 2 and completes the process of meiosis. And I've got 2 cells here. You can draw both of your cells, both of the cells going through meiosis 2 in your notes. I'm probably just going to draw one of them. So I'm going to follow this cell as it goes through meiosis 2. In meiosis 2, think about this as the phase in which you're trying to get rid of the replicated chromatids. It's a lot like mitosis. There is no pre-meiosis 2 interphase at this point. The cells that have just completed meiosis 1 go right into meiosis 2. And prophase 1 would be the first step of meiosis 2. In prophase 1, all sorts of things can happen, like the genetic material can coil up again. If it was uncoiled, it can coil back up, so the chromatin coils up again. I didn't even draw that in my cell over there that it was uncoiled because I kind of want to see my chromosomes. So the chromatin coils up to form chromosomes. All right. The nucleoli, if there are nucleoli, which there probably are, I didn't draw them in, disappear. And the nuclear envelope ruptures late in prophase 2. The spindle reforms, you know, that spindle that we had over there, the spindle forms so that it can start moving chromosomes around, right? So we've got a spindle forming, you know, so like that. We've got the nuclear envelope rupturing, and we have 2 chromosomes in this cell that are now visible. One long one, one short one. And now that cell is ready to go through metaphase 1. I'm going to see, metaphase 2, sorry. I'm going to see if I can fit it all on this one, with this one slide. We'll see. In metaphase 2, the spindle attaches to the chromosomes. So let's write that down. Spindle attaches to the chromosomes. This time, the spindle is going to attach so that sister chromatids are attached to fibers from the opposite pole. So if you've got one chromosome here, spindle fibers from one pole attached to one of the chromatids, spindle fibers from the opposite pole attached to the other chromatid. And then due to the action of kind of this tug of war that happens with the spindle fibers and the chromosomes end up aligned in the midline of the cell or aligned along the metaphase plate. So the chromosomes align along metaphase plate. But they are not with their homologous partner anymore, are they? There is no homologous partner here. So they line up single file. In this cell, we have just 2 chromosomes, right? A long green one and a short red one. Their arms can kind of be wiggling around, but they line up in the middle of the cell, or in the midline. Okay. What's going to happen next? Anaphase 2. And no surprise at what happens in anaphase 2. Draw your cell long and skinny because the cell elongates due to the action of the non-kinetochore fibers. I'll draw those in a second. And what else happens? Sister chromatids separate from each other and move toward opposite sides of the pole. Sister chromatids separate and move toward opposite sides of the pole. Opposite sides of the cell, or opposite, let's just write opposite poles, that'll be good. Let's draw that in a little bit. So now we've got, the single, I guess you would call it a full-fledged chromosome now, but the sister chromatids separate and are moving kind of in these weird V shapes toward the opposite poles. They're being kind of pulled, or they're walking toward the opposite poles. And then the cell elongates due to the action of those non-kinetochore fibers that are pulling it apart, all right? This is looking a lot like mitosis at this point, except we have only one set of chromosomes. In telophase 2, in telophase 2, all sorts of good things happen. The spindle disappears. The nuclear envelope reforms around those chromosomes. So let's write these down. The spindle disappears. Those pieces are going to be used in the cytoskeleton. The nuclear envelope reforms. The nucleoli reappear. The genetic material, the chromosomes, uncoil at this point and become chromatin. So I'm going to write chromosomes uncoil to become chromatin again. I'm not going to draw that yet. I want to see my chromosomes. And cytokinesis happens. The separation of the cytoplasm to make two cells, right? Let's draw in what we have inside, what chromosomes we have inside our cell so I can see them. All right? I have one long green, one short red in each of these cells, right? And then we end up, after cytokinesis, with two cells, okay? Wait a minute. I thought we were supposed to have 4 cells at the end of meiosis. Where did these cells will eventually become like sperm cells. Where did those other 2 cells go? Oh yeah. Remember, I was following after meiosis 1, I followed one of the cells that went through meiosis 2. This cell also goes through meiosis 2 and makes 2 of its own cells, right? Its cells would have, let's see if we can draw them. Its cells would have, what, one long red chromosome and one short green chromosome in them, right? One short green chromosome and one long red chromosome. Something like that. Like that, right? So we would have 1, 2, 3, 4 different products. They are going to become sperm or eggs, possibly, but sperm, let's just say. And each of them should be haploid, have only 2 chromosomes in them because they only have, they went from 4 to 2, or diploid to haploid, from 2 sets of chromosomes to 1 set of chromosomes. Those are all synonymous, right? And each of them does have only 2 chromosomes. That's good. And they should all be genetically identical. Oh dear. This cell sure looks like it's genetically, they should all be genetically variable. Every one of these should be genetically different because we're making all these variable sperm, right? These 2 cells look like they're genetically identical. These 2 cells look genetically identical. Are they really? What did I forget to draw in these cells? I forgot to draw in the results of crossing over or genetic recombination. If I drew those in, then this cell should be genetically different from this one. And these 2 should be genetically different. All 4 of them should be genetically different. At the end of meiosis, I started with 1 diploid cell. I should end up with 4 haploid products that are genetically different from each other. They're going to become the sperm or the eggs. And that's it for meiosis. We'll wrap up meiosis with another video, that wraps up and talks about the sources of genetic variation that come from meiosis that are found in sexually reproducing organisms. All right, thanks.